Well, we've got a pretty good crowd. I think we might as well go ahead and get started. Um, it's our
Okay.
pleasure this week to have our, I think, newest faculty member, Stephen Gazal, to talk to us. And I've asked Nick Mancuso to introduce our speaker. Um, just a reminder that next week we have Brahmar Mukherjee and her postdoc speaking from the University of Michigan. And I'm going to continue watching the uh, the waiting room to admit people as they come in. But I think that's really all I had to say for now. Nick, do you want to go ahead and introduce him?
Yeah. Hi, everyone. Thanks, Duncan. So I'm super excited to introduce today's speaker, Dr. Stephen Gazal. So as Duncan mentioned, Stephen just joined the faculty here at USC. He's affiliated with the Center for Genetic Epidemiology. And so we're all very excited that he's here and looking forward to all the methodological and scientific contributions him and his lab will be doing. So prior to joining USC, Stephen was a postdoc with Dr. Alka's Price at Harvard Public School, a School of Public Health. And during his time there, I think, from my perspective, Stephen developed a series of papers that were pretty fundamental in the field of StatGen that aim to leverage summary statistics from GWAS in order to characterize like functional and LD or linkage dependent architectures of complex disease. So today, hopefully he'll give us a little bit more insight into his previous work and maybe a glimpse into some future plans. So cool. Thanks, Stephen.
Okay. Thanks, Nick, for the introduction. I have nothing to add except that I'm like super exciting of being at USC, at like sharing my research with the biostat department. So in my talk of today, like I'm just gonna give like I'm gonna cut it in three. Like I'm gonna give like a background on my research, then I'm gonna show you like all my published postdoctoral work, and then I'm gonna show you like what I'm currently working on right now, which is like quite different, a little bit different of what I used to work on. So the title of the talk is Functional Genetic Architecture of Human Complex Disease. And so the goal of my research is just to trying to understand which area of the genome are responsible or contain variants at risk for human complex disease and traits. So when you want to study a human complex trait, such as schizophrenia here, for example, and if you want to know like the genetic basis of this disease, what you do is a genome-wide association study or GWAS. So I think like the crowd here doesn't need to, I think everyone here like knows what is a GWAS, but just to summarize, a GWAS is when you test every variant of the genome, every variant that you can either genotype or sequence with a phenotype. The results are plotted using this kind of graph, which is a Manhattan plot, where each point is a variant of the genome, and the y-axis represent the strength of the association between the variant and the disease or the trait. And so during these classes, like GWAS became bigger and bigger and highlight a lot of variants associated with a trait. So here's the example of schizophrenia, which is, I think, the first trait that reached more than 100 independent genome-wide significant variants. And what we can say, like based on this kind of results, based on this number 100 association, is that it's bringing new biological insights. We have now 100 different regions of the genome that are associated with the trait. So one of the first surprising results of the GWAS was the limited amount of information what was bring at the end by these genome-wide significant hits. So in the case of schizophrenia, if you take the signal bring by the top hits, the ones that are genome-wide significant, these explain 3% of the heritability, or 3% of the variance of the trait, and it's pretty far from the variance you can observe from twin studies, which is here 0.64. Which means that, okay, there is some more information, but we're missing a lot. And this kind of pattern is observed for schizophrenia, but has been observed like for all the GWAS. So then methods were developed to look at not the fraction of heritability that were explained by the top, but by looking at the fraction of heritability that were explained by all human common variants, and not only the ones that pass the genome-wide significance threshold. And as you can see, there's still a gap, but there is 10 times more signal in these variants that did not pass genome-wide significance than the ones that pass genome-wide significance. And so what does it mean? It means two things. So the first one is that human complex traits are highly polygenic, which means that they are dominated by a lot of common variants with a small effect that do not pass the genome-wide significance. And second, it means that it might be of interest to develop some statistical method that will investigate all the variants rather than the ones that has been that pass the genome-wide significance threshold. So a method has been developed in my lab called stratified discord regression. To do that, I will come back later on this method. But what you can do is, rather than looking at, so you can look at all the variants to have an estimation of heritability, but you can also look at all the variants in a particular functional annotation to learn about these different regions. And so one of the most striking reasons is that when you look at coding region, and so we all know that coding regions are important because they are the ones that code for protein. So these regions contain only 10% of the variance that is explained by all common SNPs. And so this number of 10% is pretty consistent across the traits. It's not observed in one or two traits, it's like consistently observed in all the traits. So if 10% come from coding variants, it means that 90% of the signal come from non-coding variants. But what was interesting to observe is that this non-coding signal is enriched in cell type specific action. So for example, if you look at like regulatory regions such as enhancer promoters that are brain specific, you will see that these are enriched for variants associated in schizophrenia. Similarly, if you look at autoimmune disease or like such as rheumatoid arthritis, the signal will be enriched in regulatory regions that are immune specific. And so what does it say? It says, okay, like there is some polygenic signal, and this polygenic signal make actually like a true biological sense. It's not just randomly distributed across the genome. Okay, so when I started my postdoc, like I think like these results strike me, and I was wondering myself two questions. So the first one was, so all these results are based on common variants that you can have from genotype data, because all GWAS use genotype data. And so I was wondering, like, what do we expect for lower frequency variants, especially rare variants, because we're at a time where we're designing like sequencing studies. And if we want to invest in, and the question was that if we want to investigate a trait such as schizophrenia, what should we do? Like, what should we do like when we design a sequencing study, when we look for rare variants with strong effect? Should we do whole exome sequencing and focusing on coding variants, or because of these results, because we observe that really like a few amount of the signal is due to coding regions, should we on the like maybe plan to do whole genome sequencing study and really look for rare non-coding variants that are brain specific and that have a strong impact on schizophrenia? And the second question is that, okay, we have observed from common variants that we have like a ton of signal in non-coding regions that are tissue specific. And now like the next question is that, okay, but now what are the genes? And so there is a question, which is how do we connect those non-coding, this non-coding signal to kind of pathway and gene sets? Okay, so I have like two goals in this talk. So the first one is to understand the impact of negative selection on human complex trait and disease. And so what is negative selection quickly? So maybe not, I will explain that later, but I just want to show the intuition that when we plot the GWAS hits based on their allele frequency here on the x-axis and the effect size on the y-axis, we can observe first that there is an impact of negative selection, which is all variants with strong effects are rare in the population, okay? And so what does it mean is that here we might have an architecture that is different than what we observed here. And so the idea here is just trying to understand how negative selection shape common, low frequency, and rare variant architecture, just trying to understand if from patterns we observed from common variants, we can deduce the one for rare variants and deduce the regions of the genome where we expect rare variants with really strong effects. And so the second question is trying to understand how to connect functional heritability to genes. So in other words, we have like strong signals that coming from common non-coding variants, and we want to connect that to the genes. And it's not an easy question because as it's illustrated on this graph, if you have like a non-coding variant here, the target gene might not be the closest one, which is gene A, but might be like a different gene, for example, gene D, just because these genes and variants kind of interact physically, like in the 3D architecture of the genome. Okay, so the overview of my talk is I quickly introduce the method I've developed, I mean, our lab developed, and I kind of improve to partition the heritability. Then I'm gonna talk about negative selection and human complex trait, and I'll finish with strategy for linking these non-coding SNPs to their target genes. Okay, so basically, the name of the method we developed is called stratified discord regression, or SLDSC. And so the idea of this method is that if you have only GWAS summary statistics, which means for each variant that has been genotyped, the p-value, then if you're able to have some kind of reference panel with sequence data to both estimate the LD, like the correlation between the SNP, but also the effect of the SNPs that are not genotyped here. And if you have also functional annotation of the genome, such as coding, non-coding, or also some continuous annotation, such as CPG content or recombination rate, then you give that to stratified discord regression, and it gives you as an output some kind of metric quantifying the importance of your functional annotation in your GWAS. So there are like different outputs of this method. The first one is just giving you the proportion of common variant heritability explained by a binary annotation. So for example, let's say you give coding to the model and just going to tell you, okay, coding SNPs, it's around 1% of common SNPs, and it's explained 10% of the signal. And then you have like one other interesting feature, which I won't comment today, which is a conditional effect of the annotation. So as you will see, this model based by looking at multiple annotation at the same time. And so it means that if you give, I don't know, 100 functional annotation that might correlate with each other, the model will tell you the one that are driving the signal. So the ones that bring new information conditioned to all the other ones, a little bit as a multivariate regression. So I'll show quickly like two slides of method, which is something I usually do when I present to, I don't know, biostat department or like methodological people. So I'm just going to first give you the intuition behind what is LD-score regression, and then I'll explain the intuition behind stratified discord regression. Okay, so the intuition behind LD-score regression is that when you plot on a graph the mean chi-square statistics of variants as a function of their LD-score. So the LD-score is the amount of linkage disequilibrium that a SNP have with other SNPs. You will see this nice linear relationship. And so what does it mean? It means that if a SNP has no LD, what you should expect to observe is a mean chi-square statistic of 1, because all your observation will be independent. But the more LD a SNP has, the higher chance it has to be in LD with a causal variance. Okay. And so because of the architecture of human complex traits are so polygenic, you observe kind of like pretty easily this kind of pattern. And so what has been interesting is when you do the math and when you write like a simple model, when your phenotype depends on genotype multiplied by effect sizes, and you assume that the variance of your effect sizes is the same for all the SNPs, then you realize that this per SNP heritability, this coefficient tau, is actually proportional to the slope of this curve. So in another word, if you regress the chi-square statistics on the LD score, you're able to estimate per SNP heritability and the heritability of the trait. Okay, so this is the intuition, and it's pretty cool and pretty, I think, pretty simple to understand. So now what we realized when we extend that to stratified score regression is that if you complexify how you write the variance of your effect, so if you write that as a linear function of Q functional annotation, so for example, you say that the effect of your SNPs depend on the functional annotation of Q annotation, then you have like Q parameter to estimate, and these estimates can be also, this parameter can also be estimated just by extending the linear regression I introduced before to a multivariate regression. So it means that rather than regressing a chi-square statistics on one LD score, you regress chi-square statistics on multiple LD score, one LD score per functional annotation. Okay, and by doing that, at the end, you will have one coefficient for each of your functional annotation, and you will be able to deduce heritability explained by each of your functional annotation of interest. Okay, so that's all for the method. I don't know if you have some questions before I go further.
Hey, Stephen, I have a quick question. So can you, I think you did a great job giving the motivation and describing what the slope represents, but in your regression model, there is an intercept term. Can you maybe just comment on that?
Oh, yeah, sorry. Yeah, so the intercept term, okay, so the idea, like, so the first LD score regression paper was based actually like on this regression term that interests people. And so the idea was that if you have confounding in your GWAS, this should not in theory impact your slope, but this should impact your intercept, because confounding will impact all the variants independent of their LD. And so the idea is that, okay, if you have a GWAS that is highly confounded, what you will observe is the same kind of like same slope, but just pushed on the y-axis, like kind of in parallel. So that's the idea. And so these slopes help you to quantify how much confounding there is in your GWAS data.
Quick
cool,
question.
thanks.
Yeah.
So in regression, at least in traditional linear regression, once you have a fitted line, the dots are some above it, some below it. But here is almost all above it. Why is that?
Here, I think it's just because this model is not true. Like we know that this model is not true. I think I will comment that a little bit later. But this model, like if you use this model, you have a really strong bias estimates, because you kind of know, because there is a link. Okay, so this is something I think I won't go very into the details, but a link between the LD score and the true effect of a SNP. And if it's not modeled here, you will have some bias. So just in other words, like I think this is not the good model. Okay, what you observe is just there is some kind of linear relationship. It's nice, but it's not that easy. But I agree with you. Like this red line is weird. Like I think it should be like it should be above. I don't know. I did not generate this graph. I took it from the publication. But I agree with you. Like maybe I'm expecting like the red line. Yeah. Yeah. I don't know. I think there's something that might be wrong with this graph.
Okay, thank you.
Okay. 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 Okay, so now I'll talk about the impact of negative selection on human complex traits. And so before going further, I'm just going to quickly say what is negative selection. So it's written here, negative selection is a negative pressure on allele frequencies of mutations that reduce fitness. And so it's illustrated like in this like naive cartoon. Like let's say you have a new variant that appear in your population, and this variant is neutral, which means it does not impact fitness. Then these variants can become extremely frequent in the population just because of genetic drift across generation, right? So, on the other hand, if you have a deleterious variant, so it means a new mutation appears, it reduces your fitness, then negative selection will constrain this variant to remain at a low frequency in the population. Okay, and of course, like this threshold will depend on how deleterious your variant is. Okay, so now it has been like suggested that negative selection impact complex trait, and the best way to visualize that is to look at this plot, right? We have observed that all variants with super strong effects have a low frequency in the population. So it means that negative selection is acting on the trait because it constrains those variants to remain at a low frequency in the population. And so now the question I try to answer during my postdoc, where like the first one, does negative selection impact common variant disease architecture? Or in other words, does negative selection act on variants that became frequent in the population and that only have a tiny effect on a complex trait? And the second question I try to answer is, in which region do low frequency variants have stronger effects? So do we observe like stronger effect sizes in coding regions? Do we observe stronger effect sizes in cell type specific non-coding regions? So these are the kind of questions I asked. I've tried to answer using polygenic analysis by using stratified score regression. So I'll go on the first question super quickly. So what we did to answer this question is we partition heritability across a lot of functional annotations that are related to negative selection, such as the age of a variant, the recombination rate, the diversity, the nucleotide diversity around a variant, some measure of background selection around each variant. And so what we observe is that patterns pretty consistent with the action of negative selection. So for example, we observed that the youngest 20% of common variants, so in other words, the common variants that appear the more recently, are more enriched in heritability than older variants. The difference, there's like a 4 times fold difference. And so why is that? If you want trying to understand, I think it's just because when a variant is recent, this variant is more deleterious just because selection had less time to not remove them, but to decrease its frequency in the population. And so what we did to validate the fact that all these annotations were, like all these enrichment patterns were consistent with the action of not with negative selection and not another type of selection, such as, I don't know, balancing or positive selection. So we did evolutionary simulation using like an out of Africa demographic model. And so these were simulations where we were able to control the age of the mutation, their selection coefficient, and all kinds of parameters. And we observed like really like super close patterns in simulations with negative selection and in real data. Okay, So in this project, we kind of introduce in stratified correlation model a lot of annotations that were related to negative selection. So we had like this 6 annotation plus some annotation based on the allele frequency of the variants. And so a question we asked was, okay, we have this model to model the effect size of a complex trait. We call this model the baseline LG model, and this model include all the annotation related to negative selection I mentioned, plus some functional annotation. And so what I wanted to do is just to compare this model, just trying to other models, just trying to understand, like, if you model negative selection, how better do you improve your fit to the data? And so to do that, we just, I don't know, took a real genotype phenotype data and compute a likelihood of a different heritability model. And so to make like a very, very long story super short, like, so what we did is that we compare the likelihood of different model to the likelihood of the LDAC model, which was at that time the model, like kind of the state of the art model. And so what we observe is that when we use only our annotation related to selection, we have like a significant better likelihood than the LDAC model, and that we include both selection annotation and functional annotation. We also improving our model. And so this notion of model like is super important in your field because it's kind of, it's used to estimate heritability in heritability studies. It can be used also as a prior to compute polygenic risk score or to perform time mapping. And so since this paper, we've used this baseline LD model in like all the projects related to this area in my postdoc lab. Okay, so now we have shown that there is pattern of negative selection on common variant. The next question was that, okay, if there is negative selection here, does it mean that from patterns from common variants, can we predict which regions of the genome can contain rare variants with really strong effects? So of course, we cannot observe with enough power the effect size for rare variants because the sample size is not quite there. But at the time of the study, the sample size was there to look at low frequency variants, which has variants with male frequency between 0.5% and 5%. And why did we have like the power to do that? Is because UK Biobank just released well-imputed data for nearly 500,000 of individuals, which gave enough power to look at what's going on for low frequency variants, but also to partition heritability. So what we did here is that we kind of modified stratified Z-score regression to be able to compare enrichment of functional annotation in low frequency variants and in common variants. And so this is this was like the main results of this study. So each point here represent functional annotation, an important functional annotation of the genome. On the x-axis, we have the heritability enrichment for common variants. So heritability enrichment means, for example, if your annotation has 1% of the SNPs and explain 10% of the heritability, then you have an enrichment of 10. It's just a ratio between what you explain divided by the fraction of SNPs in your annotation. Okay, and on the y-axis, it's exactly the same, but by looking at low frequency variants. Okay, and so I've used the log scale here, and I only highlighted annotation for which we had a significantly higher enrichment for low frequency variants than common variants. And what we observed that all annotation with higher, significantly higher low frequency variant enrichment were annotation related to the coding area of the genome. So the most striking results was reached for non-seamless variants. So non-seamless variants explain only 2% of common variant heritability. So it's consistent with the fact that really few hits highlighted by GWAS are non-seamless variants. And so few hits are like easily like interpretable because the variants, which are like the easiest one to interpret, are non-synonymous. But we observed like nearly 10 times, like 17.3% of the low frequency variant heritability is explained by non-synonymous variants. So it means that there is way much more signal for non-synonymous variants in low frequency variants, and we expect like even more for rare variants, in these annotations. What we observed is that we observed like no non-coding annotation for which the enrichment was significantly higher with low frequency variants than common variants. And because I said it might be due to the fact that non-coding annotation are important in a cell type specific fashion. So in another like analysis that I'm not gonna show for time constraint, what we observed is when you look at pairs of cell type specific annotation and trait, what we observed is that we only observed like strong higher low frequency variant enrichment for brain related annotation and traits. So for example, neuroticism and I don't know, some brain regulatory annotations, which might suggest that there is like a higher strength of negative selection in area of the genome that are connected to the brain. Okay, and so in this project also, we did some evolutionary simulation to try to connect the patterns we observed to negative selection. And so what we observed is that the common variant enrichment, so what you observed is common variant, is only related to the proportion of deleterious variant you observed in your annotation and is independent of the strength of annotation. So it's kind of tagging the probability you have for a variant just to be deleterious, but it says nothing about the strength of the deleterious effect. And the strength of the deleterious effects can be observed by looking at low frequency variant enrichment. And so we observe that the ratio between what we observed for low frequency variant enrichment and common variant enrichment is pretty linear with the strength of selection in the functional annotation. So in other words, what does it mean? It means that if we need to design some sequencing study to look for rare variants with strong effect, so it's a good approach to do whole exome sequencing because this is where the selection has been the stronger, and this is where we expect variants with strong effects. But also we observe some kind of non-coding annotations that are brain-specific non-coding annotation, for which we also observed a strong differences. So it means that for this type of disease, there might be some interest to go to whole genome sequencing rather than whole exome sequencing. Okay, so I'm gonna stop now talking about the impact of selection on complex array, but I just want to mention that there are like also like a lot of questions related to these areas that can be pursued. So the first one is trying to understand like the different patterns of negative selection in human disease. So for example, we can ask if some traits have specific patterns of selection. And so for example, we observe like really distinct pattern of selection in autoimmune disease. It's very likely because it's highly correlate with resistance to infectious disease, which has been the phenotype that kind of shape the human genome. And we also observe different patterns in psychiatric disease, which might be due to higher strength of negative selection, like a higher, because psychiatric disease are like highly related to fitness. There are also questions about the directional effect of negative selection. Is it more like directional selection or stabilizing selection? There are also a lot of questions to be asked about rare variant architecture. Like, are the patterns like different from the one we observed with low frequency variant? And I think like this question will soon be able to be answered by looking at sequencing data from UK Biobank. And I think also there's like I think a great area to look at is look at how regulatory landscape differ across population, because it's known that adaptation like really impact your way to respond to your environment, and so it really impacts your regulatory elements. So what will be interesting to look at, and which has never really been done, is just trying to look at which regions of the genome are like differently regulated in different population. Is it related to adaptation? Is it related to the different effects we observed in GWAS of different population? I don't know. I think this is like a great area to pursue research on. Okay. Before going further, do you have any question related to selection and complex traits?
Isn't another factor that would be really important age of onset for disease? I mean, you're looking at psychiatric schizophrenia. That seems almost designed to be ideal because it certainly has an enormous effect on reproductive fitness.
Yes, so this is something we looked at, and I don't know, the patterns were not really really different. So I think like what's popping up, like when you because the traits we're looking at, some of them are not like directly related to selection. I think what's going on is that just because of pleiotropic effects, every trait is kind of related to negative selection. So what does it mean? It means that the variants that are causal are under negative selection, but they are not under negative selection because of the trait we are looking at. They are under negative selection because of like pleiotropic trait we're not super sure about. So yes, as you said, like age as onset, I think it's important for psychiatric disease because it impacts fitness, and I think this is why we observe for these traits, in particularly stronger effect for low frequency non-coding variants. For autoimmune disease, there is something like completely different, which is genetic correlation with resistance to infectious disease, which like strong by way much like I don't know, like the strongest pattern observed for negative selection observed in autoimmune disease, rather than its young onset, such as Crohn's disease or late onset as rheumatoid, something like pretty consistent. So yeah. So to answer your question, yes, this play a role, especially like for psychiatric disease, but there are also like, it also impacts disease with late onset just because of pleiotropic effect with other traits.
Hey, Stephen, I had another quick question.
Yeah.
So, you know, when you're, measuring the evidence in favor of like selection existing for these traits as a function of minor allele frequency or allelic age, what are your thoughts on like the impact of drift and how that is, maybe there may be larger impact of drift like for common alleles versus rare, and like does that have any effect on how estimates on just like inference in general under this model? Because if something's still new and rare, I don't know how much drift there is compared to if something's common. There's
yes, I think what's going on for common is that it's deleterious, but it's tiny deleterious, and I'm not really sure that selection is designed to remove this variant from the population. I think it's just, I think at some point you just accumulate in your population common variants with low, like that are like tiny deleterious.
Gotcha.
Maybe to take into account drift in the model, I don't know, like, so to match our results on picker results, like theoretical result, we did some kind of evolutionary simulation using an out of Africa demographic model. So I kind of feel like, like drift is kind of taken into account, like in this kind of theoretical model.
Yeah, right. I would expect drift to be part of that. Okay, cool. Thanks.
Okay, so now I'm kind of, so I have like 15 minutes to talk about my ongoing work on trying to linking disease SNP to genes. And so when we were to say like, where is the field right now? So our field has been like pretty good to make inferences about what I call the variant architecture. So now we're pretty good to know like what is the causal variant of a hit using like some kind of fine mapping studies. We're pretty good to know what type of variants are important. For example, enhancers that are cell type specific or recent variants with methods such as stratified discovery regression. And we have also some estimates of how many variants are causal. So there are methods to estimate polygenicity, and now it doesn't surprise us anymore just to say there might be around 10,000 causal variants for a disease. And so I think now where the field is going, I think it's just trying to ask the same question, but for genes. And so this is what I call gene architecture. So for example, when we have a causal variant, we want to know what is its causal gene, what is the regulated gene. What we don't want to know anymore is that what type of variants are causal, but what type of genes, so like which pathway, which set of genes are important for a disease. And I think at the end, like one important question is that how many genes are involved in a trait. And I think it's important just to know first, like when to stop do GWAS, like when are we happy with GWAS? Is it after like detecting like 10, 100, 1,000 genes? And as you can see, like if we say that there are 10,000 variants that are associated with a trait, it's kind of okay. But saying that 10,000 genes are associated with the trait, it's kind of not okay because it's half of the gene. It's kind of pretty clear that it's not half of the genes that are involved in a trait. So I don't know, like all these questions are kind of important to answer. And as I said, it's super hard because the target gene may not be the closest one, as it's illustrated on this graph. And I think it has been illustrated pretty nicely also in this famous example, like which is called like the FTO example. So FTO, like so for many years there has been like a hit in type 2 diabetes and obesity in an intron of the FTO gene, and for many years like people thought like FTO was the causal gene. But what happens is that this paper suggests that the causal gene, the target genes are not FTO, but IRX3 and IRX5, which are more one megabase away. And to prove that, they use like a lot of evidence, and two of them come from functional data. So first, they used using high-C data, which is a type of data set looking at the 3D architecture of the genome, that the causal variant is in 3D interaction with the promoter of IRX3 and IRX5. And then by using gene expression, they show that the genotypes of the causal variants mediate the expression of IRX3 and IRX5 in a relevant cell type. And so now it's pretty clear that the way we're going to connect GWAS hits to their target genes is by leveraging evidence from functional data. And there has been like a lot of functional data that has been generated to do that. So the most well-known is gene expression, but there's a paper showing that gene expression is not enough to explain everything. And there are a lot of algorithms that has been published to, for example, connect an enhancer to the gene it targets, or to use high-C data to connect an enhancer to a gene, or just to generate some promoter capture high-C data, which is a type of high-C data that only look at the loops with the promoter of every gene. And so now we have like several questions like regarding like all these methods. The first one is that how accurate are these strategies? Like if for the same SNP is linked to two different target genes using two different strategies, which strategy should I the most be confident about? A second one is which fraction of disease SNP gene pairs does a strategy detect? Maybe it's only 1%, maybe it's 10%, we don't know. And the last question is how could we combine this strategy together just to aggregate evidence of all these independent strategy together? And so the challenge to do that is there is like a really limited set of known causal variant target gene pairs. So there's like a few examples of cases where we have a fine map SNP and we know the target gene for a given disease. And there is some kind of list but this list is often kind of biased toward like a causal SNP, which is super close to the target gene. And so what I'm proposing here is just to use polygenic analysis of disease heritability to solve these questions. And so what are my ultimate goals here? So my ultimate goal is just to kind of build a SNP2 gene, or this is something I call S2G map, aggregating functional data sets, so that for a given SNPs, we'll be able to score all the surrounding genes with some kind of probabilistic score, which will depend on the functional annotation we will have. Okay, and then when we have this in hand, there will be like multiple applications we can think about of, and the one I'm going to talk at the end is how to use this kind of map to rank genes implied in the disease. Okay, so I'm just going to speed up a little bit because I have only 10 minutes left. Okay, so briefly, what is a SNP2 gene strategy? So it's a strategy that for a given SNP give you some kind of target gene. So it could be either gene A, it could be gene B. Often what's going to happen is that for a given SNP, you have like different scores for different genes. And so a lot of my work has been for many strategies convert these scores to probabilities. So there are like different type of SNP2 gene strategy. I won't go like very into details in all of them, but just to say that there are like different kind. So the first one is what I call like the confident SNP2 gene strategy. So for example, these are exon and promoters, and it's that strategy like when you have a variant in an exon or in a promoter, you're pretty confident about what is the target gene. Then you have the naive SNP2 gene strategy, and also it's just like if your SNP is in a gene body, you take in the gene, in a gene plus minus some window, you're linking that to the gene. Or sometimes you link that to the closest TSS. Okay, then you have strategy based on gene expression. So you can either take all a GTEC CCQTL or do some fine mapping to reduce the number of SNP you're linking to the genes. There are also some methods looking at trying to link an answer to their target gene by looking at correlation across cell types. You look at the correlation with the present absence of the enhancer on like multiple cell types, and you're trying to correlate that with gene expression just to see if you can see some kind of correlation. Then you have like multiple strategies based on high C data, so based only like on the physical interaction. And I don't know, you have like also like different type of strategy, but like I would say like the four main strategies are like the confidence, the naive, the expression, and the high C strategies. Okay, so now what do we want to know for each of these strategies to be able to kind of measure their accuracy? So I've decided that there's like four different things you want to know. So the first one, you want to know what is the fraction of causal SNPs that are annotated. Okay, and this is pretty easy. It can be estimated using stratified discord regression as a fraction of heritability explained by SNPs linked to all genes. So for example, coding regions will have 10% of the causal SNPs that will be annotated. And for closest TSS, it will be 100% because 100% of the causal SNPs will have a closest TSS. Okay, the second question is how precise it is, or in other words, what is a fraction of annotated SNPs linked to the correct target genes? And this is kind of hard to estimate, and I'll show in the next slide the trick we developed to estimate the precision of a SNP2 gene strategy. The first question is what is the fraction of disease SNP gene pairs that has been detected? And this is like the recall definition. And the recall can be estimated just by multiplying the quantity 1 by quantity 2. And the last question is what is the best strategy? And to do that, we use like the F1 score, which is like a classical metric used in classification, which is the geometric mean of the precision and recall. Okay, so when we see that, we can say that, okay, so the only piece we're missing is precision, and when we have precision, we'll be able to have like a clear, better understanding on both SNP2 gene strategy. Okay, so it's pretty hard to, I think it's nearly impossible to directly estimate the precision of a metric. So the trick we developed to do that is we made the hypothesis that if we have a SNP with a high effect sizes, this SNP is more likely to be, no, we expect a precise strategy to link this SNP to a gene which is more critical. Okay, so in other words, like if you have some a priori on some set of genes that are important, and I'm going to call like this set of genes critical genes, and there will be genes for which you have evidence from exon, then a precise strategy should link SNPs to with high effect to these critical genes rather than genes that are not critical. Okay, and so as we can see here, like if we have a strategy that link these two SNPs with high effect to the gene that is critical, this strategy should be more precise than the last strategy, than link the SNPs with high effect to gene B. Okay, and so the idea is that when you aggregate all the signal across the whole genome, what you have is that you have some kind of precision definition, which should be a function of the heritability explained by SNPs linked to the critical genes, divided by heritability explained by SNPs linked to all genes. So in other words, you want to maximize this ratio to have the most precise strategy. Okay, I'm going to skip now like the details to go from this to a precision value, which is between 0 and 1, but this is something we kind of working on. And so very quickly, here are the results of the accuracy of SNP2 gene strategy. So on one hand, you have exon and promoter that are super precise, but that have a low recall. On the other hand, you have like closest TSS, which has a high recall, but like a pretty low precision. And in the middle, you have like the best strategy, such as an answer from roadmap that has been linked to the gene using gene expression. You have FindMap CQTL, a method called ABC, and closest TSS when you constrain the distance to be pretty close to the TSS. Okay, and by using the F1 score, we can rank the different strategies. Okay, now I'm gonna speed up a little bit. Okay, so I think I'm running late. Okay, so now the next step we're trying to do is now we have a function we can maximize, which is the F1 score. We want to maximize the F1 score of a strategy. We can try to, we have designed a framework to build a combined strategy, which is a strategy that uses all the SNP2 gene strategy. And for each of the strategy, we'll have some weights. And these weights will help us to kind of build a combined score that will need to be converted into some probabilities after that. But the idea is that when you build your F1 score function, you have a function where you can estimate the weights to maximize this function. And so, in other words, like now we developed a framework to estimate weights that maximize the F1 score. And so what we can see is that by doing that, we can like pick the best SNP2 gene strategy, give different weights to them, and give a strategy that kind of maximize the recall while having a pretty good precision. Okay, and I think I'm running out of time, so I'm gonna skip the application on gene ranking, but very quickly what we did is we combine these this new map to find mapping algorithm just to try to estimate how many genes are involved in human complex trait. And so what we observe very quickly is that 40% of the heritability can be linked using our method to genes. And when you look at this 40% of the heritability, around half of it comes from the top 1% of the genes. Okay. So to quickly conclude, so I've shown you like a method, statistical regression, to partition heritability across functional annotation from Biostatistics statistics. Then we have shown you in the first application of statistical regression that common variants are on the negative selection, and that low frequency variant architecture is impacted by the strengths of selection in functional annotation. And second application, I've shown you like a new framework to evaluate and combine SNP2 strategies. And in my quick application, I've shown you that 1% of the genes explain around half of the signals that could come from the genes. Okay. And I'm going to use my last seconds to thank my postdoctoral advisor, Alcus Price, all my colleagues, and just to mention that I have a new lab here, and we try to recruit grad students with computational background who are interested to work on this kind of questions. Okay, that being said, thank you for your attention.
Yeah,
Happy to take the question.
Yeah, I know there's a class that's about to start, so I think some folks may have hopped off, but you know, I think if anyone has questions, why don't we contact Stephen? Do you have your lab website or email address?
Yeah, Gazal at usc.edu.
Okay, okay, fantastic. That excellent talk, and
Okay,
very excited to have you here. Cool. Thanks, everyone.
Okay, thanks everyone. Bye.